studying the leatherback sea turtles. And what I'm going to be doing is I'm going to be describing shell ossicles of a specimen that I have that was found out in Rancho Santa Margarita. And we think it's about 15 million years old from the Miocene because it was found in the Kanga Formation. So I'm going to be describing the ossicles, which is where all of the definitive characteristics of a leatherback sea turtle are found. This specimen is important and this research is important because there haven't been any specimens from Southern California or actually the West Coast at all that have been described. We have specimens from the East Coast and from other parts of the world that have been described and fit into the evolutionary timescale, but we don't have any from California or from the West Coast.
Hi, I'm Vreeland, and I'm here talking about my master's research project. I'm doing work at Fullerton, and we are working on tying in with the biology department here who's working on oyster restoration off the coast of Orange County. Right now there's only one native species of oyster, so we want to help them figure out why there's only one native species, how long has there always been one native species here. So we did some sampling in Newport Bay on some Holocene oyster beds, and we're also doing some work in the Cooper Center, which is working in conjunction with Cal State Fullerton and OC Parks. And we're going through their collection there and looking at the oysters throughout Orange County and seeing if there's only been one native species or multiple species that live here. So that's my project.
So my name is Beth Malenick, and I'm working with Nicole Bonoso, and our project for my master's thesis proposal is deciphering biotic responses to rapid climate change at the Plio-Pleistocene boundary. So what we're basically doing is seeing how marine communities change during a recent climate change period. So about 2 million years ago, the Isthmus of Panama closed due to plate convergence. So when the isthmus closed, we saw a change in sea surface temperature, nutrients, and upwelling. So all of this kind of leads to the stability of a marine community. So when it was disrupted, we saw a lot of instability. So what happened was we had this noticeable extinction event during this time. Most of our research on this time period comes from the Western Atlantic. And what they find is that, yes, there was a significant extinction event, but we don't really see a decrease in diversity. So even though we have organisms dying out and going extinct, recovery rates were really tremendous during this time. So what we're doing is kind of the first study of its kind in the Eastern Pacific. So we're going to be looking hopefully at 2 locations, one in Brea and one in San Diego, to kind of get our samples out. So this summer I'll be identifying organisms for many of hours. And then we are also going to run a stable isotope analysis on some of our samples to kind of further pinpoint temperature changes. So we're looking for more specific seasonality results. We already know it got colder during the Pliocene, but we want to know if there were warmer winters, cooler summers, so something like that. We're also going to be working on using an ICP OES to kind of, we're going to dissolve our shells and kind of hope that it will tell us just generally what kind of isotopes were in the shells at the time. So we're going to kind of equate that to nutrients. So the whole purpose and the whole significance and why I think this project is really cool is most of the organisms we're going to be looking at during the Pleistocene are still alive today. So there are modern fauna. And we can kind of take what we see them doing in response to this climate change 2 million years ago, which is still recent time, and kind of use that as a proxy to what they might do when we see the really dramatic shifts in climate due to global warming. So that's the basic, big picture with my project. We're really excited. Hopefully we'll have some results by next spring. So that is my project.
My name is Mihai Ajou, and my topic is in invertebrate paleontology. We're going to go out and study a sponge reef mound that was discovered by Dr. Bonoso. We're going to document and look at the fossils within this mound and describe them and its constituents. Our location, our site, will be in South Canyon, West Central Nevada, and we're going to go out during this July, June, either July or June period, and spend about a week checking out this sponge reef mound. And, what a sponge reef mound, prior to, in the Paleozoic, the primary reef builders were Rugos and Tabulet reef builders, and then you had the end Permian mass extinction where like 98 or 96% of all marine life died off. And then you have this period where there's a gap where you don't see a lot of reefs, or you see just like microbial mounds. And Dr. Benuso found that this sponge reef mound that's new. And not a lot of information, there's not a lot of paleontological information about sponge reef mounds from North America. There's some information from Europe, but not a lot from North America. So we're going to document this sponge reef mound. We're going to use a grid rock sampling method, where we put out a grid on the area, the mound, and we're going to extract rocks at every grid intersection. And then we will polish the rocks and inspect them for macroscopic fossils, see what's inside making this sponge reef mound. Because there might be other organisms within between. We're also going to describe the rock within. And we're also going to make thin sections and check out this sponge reef mound to see what organisms are within. We're going to use a point count method to randomize and get some statistical data and tally what organisms we find within in the invertebrate fossil sponge reef. So our goal is to document whether any macrofauna play a limited role in the reef structure. And my hypothesis is I think that the reef cements are the main constituent of these sponge reef mounds. This is our paleogeographic map here. It is of the late Triassic, 242 to 228, Medinian to Carnian age. And this is our map of the site, if you can see here. And here's a topographical map of where the sponge was found. So that's what I'll be doing this summer.
My name is Elizabeth, and I work with Dr. Adam Woods at Fullerton. My focus is using trace element analysis to gain insight into how primary producers recovered following the end Permian mass extinction and how their recovery relates to oxygen levels. Okay, so basically I took 45 samples at varying depths within my core, and I analyzed them using ICP-OES. And then I compared my values to the values of the world average shale to get enrichment factors. And I looked at 3 different geochemical suites. One is productivity indicators, another is sedimentology indicators, and paleoxygenation indicators. And by looking at the relative enrichment of my values to the world average, I could say that I have high productivity and anoxic but not eugenic conditions during the deposition of my core. So the conclusion that we made from this is that there was a robust recovery of primary producers following the end Permian mass extinction, and that anoxia was still persisting during this time. So my time scale is the lower lower Triassic, the Grisbackian age, and the core is a very finely laminated black shale from the Mani Formation, which was deposited in the northwestern periphery of Pangea.
My name is Cortez, and my focus is paleontology. This particular poster has to do with invertebrate paleontology, and it actually has to do with the recovery after the Permian Triassic mass extinction. And we basically took samples from a location in Nevada that was once part of the back arc of the Sonoma orogeny. And we kind of went up in 10-meter intervals and took samples at each interval. After we took samples of each interval, we actually came back and made them into thin sections. From the thin sections, I was able to do point counting and actually come up with abundancy counts, and these are rough estimates on the percentages of the actual abundances that I came out with. The the interesting thing about the abundances that we actually came out with was the presence of foraminifera. These are some pictures of the actual forams that we were able to find, found within the thin section. And forams can tell you a lot about what the environment was like in the oceans at that time. So if we're able to identify the forams down to species level, we're able to kind of figure out what's going on. Because we do have this increase in bivalves at one point of the strat column, when we have a very slow decrease of abundances in bivalves, and at the very top of the column we do have a 10% bivalves, which means it's increasingly low than we did before. But as we actually see the bivalves decreasing, we actually see the increase of foraminifera. So we kind of can see that maybe the presence of forams have something to do with what's going on with the bivalves, but we have to go back and actually do some more work to see if that's what's going on.